All right, folks, we are going to get kicked off with genomics in just a moment here. For those of you who have not yet joined in a session today, I wanted to make a couple of quick reminders. All participants are muted and have camera off by default. When we arrive at the end of a talk, the speaker will be able to call on people to ask questions. If you would like to ask a question, you can raise hand using the raise hand icon at the bottom of your screen. And that doesn't disrupt the speaker in any way. So if you wanted to raise your hand while they're still speaking, that's totally fine, but we will hold those questions until the end of the period. And you are welcome to either, when we're in that Q&A period, to unmute and ask your question, or if you're feeling shy, you can also put your question in the chat and we will read it out loud so that it's recorded for when people look at the talk later. All right. Well, it just turned over to the top of the hour. And so without further ado, we will go ahead and get started with Jose Martinez speaking to us about classification of moths. Take it away, Jose.
Let's see. You can see it, right? Perfect. Hello, everyone. My name is Jose Martinez. I'm a researcher at the Radio Museum of Natural History. What I'm about to show you guys is what I've been doing for the last 7 years of my career. So, oops. I'm working at the museum as a curatorial assistant and also working on the Noctuoidea superfamily, the largest superfamily in Lepidoptera. I'm working with, especially with nootropical species, but for my PhD, part of my career now, I'm working in other groups not related with the nootropics. So for those that are not familiarized with Noctuoidea has 7 families now. I have 6 here, but currently they found a new family in Africa, and it's thanks to one of my friends. So this group is one of the worst groups to work with, because everything in there is really a huge taxonomical or systematic problem that has been for decades. We tried to fix this problem, and this year we published this paper. Unfortunately, we didn't have enough data to change things different from the previous work. So that's actually one of the problems we have with the reviewers in the beginning. But still, we have the huge justification that Noctuoidea is monophyletic, right? So also we could identify some synapomorphic characters that we can use to identify Noctuoidea and corroborate the monophyly of this group. So we use the tympanoid organs. Tympanoid organs are the most common thing to see in Noctuoidea adults in general. And this is because they are in the metathorax and the first segment of the abdomen. So this is a very common thing. No other Lepidoptera have these characters in general. So another important thing is larvae, we use chetotaxy and to identify these characters, and they are unique. So they have this character, and no other Lepidoptera have this. The problem became when some of the people working in Noctuoidea, they decided to stop using morphology and just work in genomics, right? So despite that, I understand that genomics is the next thing that we're using now. It's still just a tool. So it's not the whole truth, right? So people that stop using morphology, this creates a lot of problems. and generating more gaps instead to bring more light to these problems. And this is because every single Lepidoptera family have apomorphic characters. So some of the actual sphinges, they have this caudal spine. So Saturnids, they have these huge bodies with secondary setae. Laciocampis, they have these secondary setae like hers. You know, all of them have characteristics that you can actually identify. Problem is when you were start working in Noctuidia, they don't have anything. So that's one of the main problems in there. So what I decided to do, because it's a huge family with 72,000 species, it's impossible for me to work on that. So I decided to work in a small family, and it's called it's called subfamily, that's called Pantaeni. But Pantaeni is basically is going to be the pinpoint for my research. So, and I explain why. So when I started with this group, it was to try for 44 genera and 150 species that were included in the beginning of the subfamily, right? So it got in the point that was doing the morphological part for this, and I found that this group is totally different from the rest of the other groups, making more easy to understand that this is actually not subfamily, but a family. So obviously this is my specialty is in morphology, right? But I'm not against to use other approaches. So that's why I started using genomic data, right? So I started using anchor hybrid methods, using the Lab1 prop kit that's been used in the was proposed here in the Florida Museum of Natural History, where I used 113 putative Noctuidae. So I decided to work just with the Noctuidae family because it was very hard to work with the entire thing. And then I add 62 Noctuids from the other families just to place the group that I'm working in the tree is properly placed. Sorry. And 25 specimens from all the Macroheterocera and 3 butterflies. It's actually just to justify how deep is the analysis. So for the analysis that I used was the normal maximum likelihood analysis using IQ3, and just to corroborate the genera that is close related, we use multi-coalescence analysis. So I examine morphological character from immatures and adults, to be exact, 235 genera and 309 species. So at the end of the results, I could find that mostly of these families in Noctuidae, they were actually families. And it's based on not just morphology, but also the actual support in the phylogenetic analysis show that they are actually different things. But there was 2 other clades in this area around here, which includes the group Bahisaridae that is not there yet, because they have really low support. So based on this, I decided to redo the analysis, and this time adding more data. And this very time, I'll actually add the new family from Brian and Lauren, and I use exactly the same team method, exactly the same outgroups, and I could identify that not just the 6 families that these 5 families that I proposed before were there, but also there were other 2 families that were totally placed in different plates. So based on this, the main problem is like most of the people was justifying that they are in the same clade, right? And because they are in the same clade, usually say that same clade means synonymy, right? But here I have two clear examples that's not true. So the first one is that the actual project that we did in about bombicoids, we found that this clade here have 100% support. All these families have 100% support. And people that was working in genetics and genomics, they were saying that it was actually the same family. But the experts in the group, they were saying that that's not true. So that's why and actually they split this in different families. Same thing happens with one of my friends here. He was working with tiger beetles, right? So well, they were trying to separate tiger beetles from the ground beetles. So for those that are not related with this, the groups in general, tiger beetles are it's a really big and very diverse group that is mostly terrestrial, and they actually prefer to, walk instead to fly. And the same thing with the brown beetles. The differences are not just morphologically, but also chemically. The ground beetles, they actually produce chemical defenses that separate this group from the rest of the other groups. In conclusion, what I want to say is that we start understanding the evolutionary history of noctuidity, which is still very difficult to comprehend. Separation of the noctuidity sensu stricto versus noctuidity sensu lato, which is still is ongoing because or something was only 1% of the entire noctuoidea that's in the world. Oh, sorry, it was 0.1 sampling from the entire noctuoidea now. So, which makes it very difficult to understand what is happening in noctuoidea so far. Again, close relationship always justify synonymies in general is one of the main problems that we have in general in systematics right now. I'm proposing 7 new families using genomic and morphological data. And I understand that it's important to have the molecular data, but also it's important to have morphology to corroborate this, because mostly of these really impressive trees, they don't say anything if you don't have something to corroborate what is happening. It could be morphology, it could be chemical ecology, it could be just biology or something that make them understand why the tree it looks like that. And of course, noctuoid needs a lot of work, especially in the area of systematics and evolution. For my current projects right now, I'm working on the polygenetic revisions on Fontani and Diopsy, two of the new families that I'm proposing. But also I'm reclassifying another subfamily using exactly the same methods, exactly same approaches, which is the family Erebidi. So I start with 2 subfamilies that they were families for a very long time. And for some reason, the people working on this decide to place them as subfamilies, which is not corroborated by the morphology, is not corroborated by the chemical defenses of these groups, and also is not corroborated by the genomic data. So also since Noctua is very big group, I'm working in other things not related with just systematics and evolution, also working in genomics where I'm working with the Genomus Santopastis, which is a group of moths that produce, I don't know if you guys can see this, I guess. This thing is, oh, right there. So this mod is one of the few mods that they produce, they actually present sequestration. So for those that don't know what the sequestration is the actual strategy or defensive mechanism that some moths have to sequester chemical defenses from the plants and use it against the predators, right? So I also doing some taxonomy, but unfortunately taxonomy is not very foundable right now. So that's why people stop doing that. And I prefer to do this as a, um,
Jose, unfortunately, we're gonna have to wrap up and allow the next speaker to
okay,
transition to
okay.
their talk. So you can finish speaking while their slides come up, but we are gonna have to wrap up.
Yeah, okay. Well, it was my last actual slide. That's okay. It's just showing what I was doing for my research. But thank you for it. I'm sorry to get to more time this post too.
Oh, no problem at all. All right, thank you for that talk. We don't have time for a question right now, but if you would like to ask Jose something, he will be in the session, so you can use the chat function to ask your questions. And we are going to move straight on to our next talk about hybrid zones. All right, take it away, Marina.
Yes, thank you. So I'm a researcher at University of Gothenburg, and this particular work is done in collaboration with Alan Lemoine, Rui Faria, Chesney Johannesson, and Roger Butlin. And just to start with, I thought of first explaining and give you kind of a picture in mind about a hybrid zone. So this picture on the left is an example of a hybrid zone. It's a habitat with an environmental transition, a gradient of selection pressures actually that these snails shown on the right experience. Littorina saxatilis, bear in mind the name, I will mention another species soon as well. And so in response to the selection pressure, for example, crab predation on this side closer to us, and not so much wave action, but in contrast to a lot of wave action in this area on these large rocks and not so many crabs and not at all crabs, we have actually a transition of phenotypes of size of these individuals going from very large on this side to very small on this side. And if we actually plot how these phenotypes look like over this shore, we will actually see lines like this, although this is the frequency of alleles, but it doesn't matter. Also, if you plot the phenotypes, you will also see a similar shape here. The classical hybrid zone theory actually focuses on the balance between natural selection that I just explained and migration. And what we actually have usually is sharp environmental transition at some point. And on the left, for example, we have a series of patches where small individuals are favored. And on the right, we have, for example, selection for large individuals, just to give you an example similar to what I just explained in terms of snails. And as I said, if the phenotype that is under selection, if that's what we are measuring, if we plot it as a function of this distance and geographical position, we actually see this. Sorry, there was no video of me. We actually see this smooth clinal shape. We know quite well the theory about the shape of this line here, which actually shows, what we expect in a particular position. So the average phenotype in a particular location. We also know that it's the balance, as I said, between migration and selection. So if we keep the migration rate the same but increase selection strength, we will actually see a narrower cline, so a stronger barrier to gene flow. Transition here between blue, green, and red going in the direction of increasing selection strength. You see that the narrower the cline gets. But this theory doesn't really tell us what happens with the local distribution of phenotypes. It only tells us about the mean value, the expected value in a particular location. But if we look at the classical hybrid zone theory, where we have just the balance between migration and selection and actually random mating and not any specific, very, very simple assumptions, we will actually have that in the center of the zone, you know, where this expected phenotype is actually in the middle between the two extremes at the hybrid zone ends, we actually have that the distribution is really centered around this midpoint, and this midpoint actually represent, you know, individuals with this that are true hybrids. So this is unimodal distribution or unimodal hybrid zone, and this is in contrast to a multimodal hybrid zone, let's say a bimodal hybrid zone, which is shown here on the right. We have just two clusters of individuals, one kind of representing these kind of small individuals, if we think about the snail example again, and on the right we have a cluster of individuals that are representative of, you know, the kind of crab ecotype or large individuals, but nothing in between, so no true hybrids. And so the question we want to answer is really, how do we generate this bimodality or multimodality? Why are we interested in the modality? Well, because modality is an indicator of gene flow between extreme types, so the extent of gene flow, the extent of diversity, and as I said, it's a theoretically intriguing question. And we have empirical evidence that there is actually a large diversity with respect to the modality of hybrid zones in nature, and this is a picture, figure borrowed from Jiggins and Mallet from 2000, where they actually did a survey. In the top row, you have examples of unimodal hybrid zones. Bottom row shows some bimodal hybrid zones, and something in between in this second row. But we also have our own examples from our own groups, Lithuanian research community. Actually, the first example here is Lithuania saxatari that I showed you in the first slide, marine snails, and next to them are Lithuania fabalis. They live next to each other, very similar habitat, very similar selection The second one has actually a bimodal hybrid zone. You have to turn your neck a little bit to see these distributions here. Focus on the red distribution. So they're definitely not unimodal here. It's just different phenotypes shown here. So what we want to understand is what actually explains this diversity of modality here. We have some educated guesses that prezygotic reproductive barriers are potentially critical to actually establishing multimodality, which is something that Jiggins and Mallet concluded in their 2000 study. This includes assortative mating and habitat choice, for example, but also some people would say, well, if you have a very bimodal or multimodal hybrid zone, then it's a sign of very strong divergent selection locally or globally. But actually, there is no systematic theoretical studies on the topic. So what we did here is just to extend the usual hybrid zone model the classical one that I mentioned, but instead of random mating, to actually include the possibility of having assortative mating, include genetic incompatibility, something that the classical hybrid zone model doesn't actually include, and to consider the different arrangement of adaptive loci, so collinear, which is in a classical sense what the classical model includes, but also inversion polymorphism. And why? Because, well, our snails in particular, among many other species, show that there is lots of differentiation that is locked in inversions. And also to test this strength of divergent selection. So what does that do? I will not go into the model details, just to mention that the trait that is under selection is highly polygenic. But instead, I will go straight to the results. So if we consider just collinear arrangement and we just a little bit change the classical hybrid zone model in a sense that instead of local random mating, we now assume assortative mating. And I will mention here some strength of assortative mating. What I really mean is that I have a parameter that says what is the probability of mating between these two extreme ecotypes at the hybrid zone ends in relation to the probability of mating of an individual with an individual of the same phenotype, for example. So if we have this probability of mating between ecotypes of just about 1%, even less than that, we still have that the local distribution of phenotype, and bear in mind that I will always show the distribution of phenotypes in the center of the zone, where the extent of multimodality should be most visible if it exists. But under these conditions, there is no multimodality, right? So we have to actually further strengthen assortative mating, so to have more restrictions on who mates with whom. So if we go down and we say, we really restrict mating with very different individuals a lot, meaning, for example, that we go down to the probability of mating with very different individuals to 10 to the minus 6, which is a very strong assortative mating. That's when we get very strong bimodality. But this is not interesting, right? Because this is essentially different species, and then, well, it's not strange that we are actually getting this bimodality. But now if we forget about assortative mating and instead look at genetic incompatibilities, and here in the model I allowed for genetic incompatibilities between different pairs of loci, and in total there could be, for example, not for example, but a parameter that I used is that you could have in total 14 compatibilities, but per incompatibility fitness reduction is a parameter that I change here. So if you have one incompatibility, you get a fitness reduction of 1% in this case here, for example. And as you move to the right, you get a fitness reduction per incompatibility of 10% or 20%, which is quite a lot, because if you have all incompatibilities, first generation of hybrids, after secondary contact in the rightmost panel, their fitness reduction would be 99.99%. So they barely survive, but still that doesn't generate multimodality. In essence, what we need is really mortality of F1 hybrids in order to establish multimodality just due to genetic incompatibilities. Okay, but if we now combine incompatibilities and assortative mating, we are in the domain of collinear regions, but we have reasonably strong assortative mating. And here we fix that to 1% probability of mating between ecotypes. We still don't see actually signs of multimodality. So we really need very strong incompatibilities or very strong assortative mating, which is not really what we expected. So it's really the conditions that require different species all along. But what about inversions? So inversions actually do hinder gene flow between different arrangements. So they are a barrier to gene flow. But the question is how effective they are. So now if we, instead of having collinear arrangement of adaptive loci, if we assume that there is standard and inverted arrangement fixed in the different habitat parts before they come into contact and include assortative mating, what happens then? Well, we still need a lot of a very strong assortative mating to create multimodality. So inversions do help, but not really much in this case. But now if we test for incompatibilities plus inversions, then we see that we do get multimodality in this rightmost case when we have fitness reduction of per incompatibility of 15%. So this is actually less strict condition than in the case of collinear arrangement, but still a little bit harsh. But if we now include inversions and incompatibilities and assortative mating, which is reasonably strong, for example here, that the probability of mating between ecotypes is about 1%, what happens then? Well, here it's actually something that we would, that is kind of acceptable. So if we have already here that the per incompatibility fitness reduction is about 4% only, we do actually get a very nice transition towards bimodality. And then we have a very strong bimodality when we have per incompatibility fitness reduction of 10%. So here we see that the distribution of phenotypes is being stretched like a string, and then it breaks down into 3 or 2 pieces. Just to summarize here, I actually have suggested here measure of hybrid deficit, but I don't go into details what the measure is. So here if this measure is above this dashed line, then we have multimodality. 1 is actually nice bimodality. Below the dashed line, there is no multimodality, it's unimodal distribution. So in the collinear region shown on the left, well, you really need very, very strong assortative mating, as you saw, but with inversions, the conditions become less strict. This is what I wanted to show. I will not go into details, but I'm happy to take questions. The summary is really that unlike what we thought, that it's assortative mating or just pre-zygotic barriers to gene flow, we actually do need a combination of the different barriers to gene flow, and in particular, inversion seems to actually be quite good facilitators of generating multimodal and in particular bimodal distributions of phenotypes locally. That was my last slide. Thank you for your attention.
All right, perfect timing. While Daphne gets her screen up, we could potentially take one quick question. Otherwise, Marina, I think you'll be around for the rest of the session to answer any questions in the chat. Excellent. All right, well, that is the half hour. So I will go ahead and turn it over to Daphne for our next talk in the session. Take it away, Daphne.
Hi, everyone. My name is Daphne Anastasiadi, and I'm now a scientist at the New Zealand Institute for Plant and Food Research. And what I'm going to present today is part of my postdoc on the relative roles of genomic and epigenomic variation on local adaptation in wild populations of a widespread marine fish. So just a brief introduction on what is epigenetics. Our working definition is the study of phenomena and mechanisms that cause chromosome bound, mitotically and or meiotically heritable changes to gene expression or phenotype that are not dependent on changes to the DNA sequence. So genetic mutations change the DNA sequence, however, epigenetic changes rather sit on top of it. One of the main epigenetic mechanisms is DNA methylation, which is basically the addition of a methyl group to the cytosines of the genome, and this is especially relevant in vertebrates when these are followed by guanines. So if we look at this from an evolutionary framework, we know that genetic variation affects gene expression, which affects phenotypic variation, and the environment through natural selection can alter the genetic variation. Epigenetic variation sits in the middle of all of this, can regulate genetic variation or be regulated by it. It can directly affect gene expression, and the environment can directly alter epigenetic variation and can also be altered by natural selection. So a couple of years ago, we published a review where we highlight that in order to understand the coevolutionary significance of epigenomic patterns, one important step is to study the contribution of epigenetic inheritance to adaptation. And one good approach to do this is through population epigenomics, which will allow us to test the uniformity or diversity of population responses or the predictability of responses in natural population. And this is the approach we used in this study to answer the more specific question about does epigenetic variation contribute to local adaptation to temperature? And for this, we used our model system, a widespread marine fish in Australia and New Zealand. It's called Tamure in Te Reo Maori, the indigenous language from the indigenous people of New Zealand, or Australasian snapper in English. I put here a map of New Zealand, so you can see that we have a great natural thermal gradient because New Zealand covers more than 10 degrees of latitude. So we sampled adult individuals of snapper across 11 areas that covered both the east coast and the west coast of New Zealand, from north to south until where snapper was found. So we have more than 500 samples of snapper, and we investigated DNA methylation patterns through whole genome bisulfite sequencing, where we have 11 pools of around 50 individuals per area. And we looked at the genetic patterns by low coverage whole genome sequencing with more than 450 libraries. And we used liver for the study that we sampled in the fish factories after the ship have landed, the commercial ship. So these are the results of the SNP analysis, and we see that the populations form two clusters. One is red, is the East Coast, and the other one in blue is from the West Coast of New Zealand. And we see some individuals from the East Coast in red that group to the West Coast. And actually, if we look to previous data on population genetics of the snapper, we see that actually there are two genetic clusters, one in the east and one in the west, that in the southern part of the eastern coast, the populations that are there group with the west cluster, and this is exactly what we find in our data. So if you see here, area F is the one that clusters with the west coast, and this means apart that we have two genetic clusters that our genetic data are valid since they corroborate previous findings. If we look to the epigenetic variation patterns, we have the mean global DNA methylation per area. We don't see any specific differences per area, but when we look all the CpGs distributed across the genome that we got through whole genome bisulfite sequencing, we see that the overall DNA methylation is around 80%, which is the value typical for vertebrates. However, we thought that comparing all the CpGs of the genome with SNPs that are by definition polymorphic is not valid. Therefore, we tried to find CpGs that we consider polymorphic. And to do this, we looked for differentially methylated cytosines in pairwise comparison between areas. And we have categorize them in two categories. So the first one is loose, we call them loose polymorphic CpGs that showed methylation difference of 10% with an FDR of 0.05. And these are around 400,000. And this number is of importance to us here because it compares with the number of SNPs we have for the same study. And then the third group is what we call strict polymorphic CpGs that had to show a methylation difference of 25% and an FDR of 0.001. And interestingly, what we could see was that if we are going towards the polymorphic strict CpGs, the global DNA methylation is actually decreasing. Then what we wanted to do was do the similar analysis for both the genetic and epigenetic dataset and look how the spatial distribution and the environmental factors might be affecting them. So for the genetic data, we could see that both geographic and environmental distance show a significant relationship with genetic distance that indicates both isolation by distance and isolation by the environment. When we do the same analysis for the DNA methylation data, we couldn't find any relationship between geographic and environmental distance and methylation distance. So this was a first indication that there might be an uncoupling of genetic and epigenetic variation in our system. Then we looked straight to the direct relationship between genetic and methylation distance and also the principal components from both data sets, and in either case we could find any relationship which further supports that there is an uncoupling of genetic and epigenetic variation in our system. And the next step was that we wanted to look more specifically to spatial distribution and environmental factors, how they may affect our two data sets. And we did this through redundancy analysis, or RDA. We chose 5 environmental variables after filtering for correlations and multicollinearity. And these were chlorophyll, velocity, sea surface temperature, iron, and nitrate. This is for the genetic data, and we can see that the model is significant, and iron and nitrate are the main drivers of this significance. And then we did the same analysis, RDA, for the CPGs. And we see that for the whole set of whole genome CPGs, we find that velocity is the main driver of this variance partitioning. But then, very interestingly, when we do the same analysis for the polymorphic sets of CPGs, we see that slowly from the loose to the strict polymorphic CPGs, sea surface temperature mean is rising and is at the end for the strict polymorphic CPGs the main driver of the differences. And then the next step was to look specifically for association of the SNPs and the CPGs with the temperature. We used the set of loose polymorphic CPGs because, as I said before, the numbers with the SNPs were comparable. And we used similar statistical tests to look for this association. And in this analysis, we were able to find 482 SNPs overlapping 407 unique genes and 254 CPGs overlapping 222 unique genes. And these results show that the SNPs and CPGs, that we have SNPs and CPGs that associate with temperature, and these are likely to have functional significance since they overlap with genes. So to sum up the study, just a couple of take-home messages. First of all, genetic and epigenetic variations seem to be uncoupled in wild populations of a widespread marine fish, the tamure. Spatial distribution and environmental factors influence the genetic but not the epigenetic structure of these populations. The main driver in DNA methylation profiles of polymorphic CPGs is temperature. And lastly, SNPs and CPGs associate with temperature and are likely to have functional consequences since they overlap with genes. And with this, I would like to finish and thank my 2 postdoc supervisors, Maren Wellenreuter and Luis Bernatchez, who unfortunately passed away last year, but he was part of this job, staff at the PFR that helped with the fieldwork, and all you for your attention.
All right, excellent. We have plenty of time for questions here. Just as a reminder, if you have a question, please use the raise hand function and the speaker will call on you. All right, Daphne, you're free to call on any speaker you would like to.
Someone has a good hand.
Okay.
I just have to
I
figure out.
see.
Okay, I'll ask them to unmute so they can ask their question verbally.
I've got it. Sorry.
There we go.
Hey, Daphne, great talk. So I would like to know what reference genome was used for calling out SNPs. I mean, reference genome, was it from the same place, like I think from New Zealand, or was it from was it sequenced somewhere else and then you used it?
Oh, yeah, it's the same species. It's from New Zealand. And actually, yeah, people in our lab have published it a couple of years ago. Now we have a second version. However, I have to say that the genomic resources from species of New Zealand are published, but they are under strict access because they are sacred for Maori. So if one So they are not on NCBI or anything anywhere like completely free. You need to ask for access for this. It's in Genomics Aotearoa, but it's the same species. Yeah.
I mean, yeah, I mean, even if it's, I understand it was from the same species, but I just wanted to know, was it from New Zealand or it was maybe sequenced in Europe or the US? Or since you had a supervisor from Canada, no,
No, it's
Professor.
from New Zealand.
Okay, all right.
Yes, from New Zealand.
And you don't have an information yet that how many of those SNPs are non-synonymous, which are like impacting the protein function. So you don't have that information yet, right?
No, I don't have that information yet.
Oh,
No.
okay.
Yeah.
Okay, no worries.
Thank you.
All right.
So there is Hong Mei.
Yeah, hello.
Hi.
Yeah, thank you for the talk. So I just wondered, for your fish model, are the SNPs and the DNA methylation occurs on the gene body, or like a non-coding region, or like what kind of area in the genome?
Well, yeah.
Actually, so we
most
are right at a quarter till the hour, which means we need to move on to our next talk. Would it be possible for you to put your question in the chat, Hongmei, and Daphne, you answer it there? Thank you so much. And it looks like you also have another question in the chat as well. So with that, it is time for our last talk of the session, which is Wendy Valencia Montoya. Are you ready to go, Wendy?